Hi, my name is Nate Bousted. I'm live here in Woodbury, Minnesota. I'm going to be giving you a rundown on the process of cellular respiration. So let's get started. So cellular respiration is a three-act play. Act 1 is glycolysis, Act 2 the Krebs cycle, aka the citric acid cycle, and Act 3 is the electron transport chain. We're going to be starting with Act 1, glycolysis. Glucose becomes energy. Now, cellular respiration, it's the extraction of energy from food through using glucose in the presence of oxygen. The presence of oxygen is a very important aspect to this. So, Act 1, it's a series of reactions that extracts energy from glucose, which is a simple sugar. Glucose is the most important source of energy as it drives all of the reactions that we're talking about today. Glucose, as you can see here, it's a 6-carbon molecule. And now it's going to be split into two 3-carbon molecules called pyruvates. As mentioned, this is the first step of cellular respiration, but glycolysis itself does not require oxygen. Anaerobic organisms, which don't use oxygen, can also use glycolysis. Glycolysis takes place in the cytoplasm of the cell, which, as you can see here, I didn't include all the pieces of a cell, just the important ones for today. Here's the cell. Here's the nucleus. I had to put that. The nucleus is very important after all. The cytoplasm is this jelly-like material outside of the cell, or outside of the nucleus in the cell. And we'll get to the mitochondria here in a little bit. So there's two phases to glycolysis. There's the energy requiring phase, as you can see here, the energy releasing phase, which is down here. And now we'll go more into the specifics of it. The energy requiring phase, the glucose molecule, which has a 6-carbon chain, as you can see, gets 2 phosphate groups added to it. That's these little guys right here. This makes it unstable and allows it to split in half and form 2 phosphate-bearing 3-carbon sugars. Now, this is the energy requiring phase. So we're having to put energy in. We have to put 2 ATP in. So these 2 ATP are going to be used up and they're gone at this point. That's the energy that was required. So now we move on to the energy releasing. We invested our 2 ATP. We want to get something back from this. So the energy releasing phase, each 3-carbon sugar is converted into another 3-carbon molecule, which is called a pyruvate. And I put the pyruvates in orange down here at the bottom. These reactions make 2 ATP, 2 and 2 from each pyruvate, and 1 NADH molecule. The 2 pyruvates are made, and the reaction takes place 2 times, so 4 total ATP and 2 total NADH molecules are made. To refresh our minds on the total process, glucose converts 1 6-carbon molecule of glucose into 2 3-carbon molecules of pyruvate. The net gain, as we're going to see here on the bottom, is 2 molecules of ATP. Remember that we invested 2 that were used up early in the process. So 4 produced minus 2 used up leaves us with 2 at the end. 2 molecules of NADH, and then the 2 pyruvates. And if oxygen is available, and it is when we're talking about cellular respiration, which is what we're talking about today, pyruvate can be broken down and oxidized into CO2 in the cellular respiration process. Now, to keep glycolysis going the NADH molecules that were produced need to become oxidized to NAD+. If NADH stays in the reduced state, glycolysis will stop. To get the NADH to the NADH+, there are two ways that this can be done. With oxygen present, NADH can pass its electrons into the electron transport chain, which we'll be talking about later. It regenerates NAD+ for use in glycolysis, while also producing ATP in the process. Another option, which we're not going to go into detail today, but I will briefly mention. So we're talking about requiring oxygen, but if there's no oxygen present or the organism cannot use oxygen, the organism will use fermentation to continue glycolysis to make ATP. And that is all for the glycolysis cycle. We're going to move on to Act 2, which has a lot of names. The Krebs cycle, aka the citric acid cycle, aka the lesser known carboxylic acid TCA cycle. That was a mouthful. And I don't know if I've ever said that out loud, so that would explain why I stumbled a bit. The Krebs cycle. Now, this is a cycle, as you can see, and we'll kind of go into that a little bit more. As you can see up at the top, we start with our pyruvate. It's one of the products from glycolysis that we just did. It's going to be transformed into an acetyl-CoA in the mitochondria. And the mitochondria we mentioned earlier, and now we're going to go back to it. It's these little guys right here. I colored in orange. They're known as the powerhouse of the cell. Now, the molecules, this is important to know that molecules called electron shuttles are an important part of this process. They accept the energy released by rearrangement and subtraction of carbons in the form of electrons. It's all about the electrons in this part. These electron shuttles here are NADH plus and FADH, and they transport high energy electrons by gaining electrons. This is called reduction. And then the opposite is by losing electrons. This is called oxidation. It can be slightly confusing, but gaining electrons, reduction. Losing electrons, oxidation. So the NADH, the reduced form, that is the reduced form, and then the NAD plus is the oxidized form. The FADH2 is the reduced form. The FAD is the oxidized form. Now let's go back to the beginning of the process. The 3-carbon pyruvate loses a carbon. So there's 3 here. Uh-oh, we lost one. And the acetyl-CoA is a 2-carbon. So where does that carbon go? Well, as you can see here, this little gaseous, cloudy-looking bubble is CO2. The carbon that's lost is going to be picking up 2 oxygen from the pyruvate, and it's going to exit the body through carbon dioxide, which we breathe out. So that's why I kind of made that a little gaseous cloud. We breathe it out. It's gone for now. Now that the acetyl-CoA has been formed, the Krebs cycle can start. So that kind of happens outside of the actual cycle, but it's an extremely important part. We can't do the Krebs cycle without that entering it. It's referred to as a cycle since the beginning product, oxaloacetate, which is a 4-carbon molecule, is regenerated at the end of the cycle. It changes throughout, but ultimately returns to its beginning form. To start the cycle, we need an enzyme to fuse to acetyl-CoA and oxaloacetate together to form citric acid. There's the name, the citric acid cycle, one of the 3 names that I was giving you earlier. Well, there it is, the citric acid. It's right here, 6 carbons. So an enzyme rearranges the atoms in the citric acid molecule into a new 6-carbon. So they shifted from this, these are just artist renderings, but just to show you that they shifted. And the energy released when the 6-carbon arrangement is oxidized, it causes one carbon to be removed. This removed carbon, once again, combines with oxygen to produce CO2. As you can see here, we're going to lose that CO2 again. Some of the energy in the form of electrons is captured in the NADH high energy compound. So the NAD plus receives these high energy electrons from reduction through the carbon molecule being oxidized. The same reaction happens again. Another carbon is lost from the 5-carbon molecule now. So we went from 6 down to 5. And then we're going down to 4 now. So we're leaving a 4-carbon molecule, once again, producing CO2, and some of the energy is released and used to reduce NAD plus to NADH, as you can see here. Now we need another rearrangement. So the 4-carbon molecule is rearranged. During this arrangement, non-carbon groups are added to and removed from the molecule. And we are going to produce GTP, which is guanine triphosphate, or ATP, adenosine triphosphate. So GTP can be converted to ATP rather easily from the body. So we're going to kind of just say that they are equivalent molecules for this time being. Either one can be produced in this cycle. And also, in addition to that, we have an FADH2 is made during the rearrangement of the 4-carbon molecule. Right here. It's very easy to lose track of what you're talking about with all of these unless you're seeing it in addition to hearing it. It makes so much more sense when you're seeing it and gets in your brain quite a bit better. So the 4-carbon molecule is rearranged one last time, producing oxaloacetate, which is what we started with in this cycle, and combined with the acetyl-CoA. Once again, some of the energy is released and transferred to reduce NAD plus to NADH. Now for one turn of the citric acid cycle, we have the production of one GTP or ATP, so we're going to say that those are equivalent. We can deal with GTP. We can convert it to ATP easily in the body. We have 3 NADH. We have 1 FADH2, 2 CO2, which are lost, and we have the regenerated oxaloacetate. So the cycle can start all over again. Now the 2 rounds of the Krebs cycle, we have 1 molecule of glucose, which we started with in the glycolysis reaction. It's been oxidized completely to CO2, and its energy has been captured. As you can see, this energy is captured. Now that we have NADH and FADH2, we can move on to the high ATP yielding process of the electron transport chain. You may have noticed that we haven't produced a lot of ATP as of yet, but we will. We're getting there. It's coming. Like I said, a 3-act play. It's time for Act 3. We need the electron transport chain. Act 3, the electron transport chain. And some other processes that are involved in this is oxidative phosphorylation and chemiosmosis. So the electron transport chain. In the electron transport chain, electrons are transferred from one molecule to another, and energy is released in these electron transfers It's used to form an electrochemical gradient. I'm trying to represent that here. In the chemiosmosis, another related aspect, very important part to this, the energy stored in the gradient is then used to make ATP. That's represented over here. I'll go into a little more depth of that, but this is the entire visual representation. I'll go into a little more depth here. Now, oxygen sits at the end of the electron transport chain. It's right here, which is very important. It accepts electrons and picks up protons to form water. As you can see, tucked down here, water is produced. Without oxygen, the electron transport chain will stop running and no ATP will be made, which would be very disappointing. We're looking for lots of ATP. We need to fuel our cells. We need to fuel all the things that we do, our heartbeat, the movements, our ability to really do anything. We need ATP. It's vital to life. And the electron transport chain is a series of proteins and organic molecules found on the inner membrane of the mitochondria. The electrons are passed from one member of the transport chain to another in a series of redox, which is reduction and oxidation reactions. We talked a little bit about those earlier. The energy released in these reactions is captured as a protein gradient, which is in turn used to make ATP through chemiosmosis. It's right here. The electron transport chain and chemiosmosis together make up what's called oxidative phosphorylation. The beginning process includes the delivery. We're talking about the electron transport chain. The beginning process includes the delivery of electrons by NADH and FADH2 that transfer their electrons, turning back into NAD and FAD. NAD plus and FAD, which can be reused in other steps of cellular respiration. Those electrons are passed down the chain. They move from a higher to a lower energy level, which reduces energy. Some of the energy is used to pump H plus ions, moving them into the intermembrane space right here. Here's the inner mitochondrial membrane. In the mitochondrial matrix as well. So as I was saying, the electrons are passed down the chain. They move from higher to lower energy level, which releases energy. Some of this energy is used to pump those H plus ions, moving them into the inner membrane space and establishing an electrochemical gradient. At the end of the electron transport chain, electrons are transferred to molecular oxygen, which splits in half. You can see here we have a half, and takes up the H plus to form water, which is very vital, as I mentioned earlier. As the H plus ions flow down their gradient and back into the matrix, they pass through an enzyme called ATP synthase, which is represented right here, this big guy right here. Now, this harnesses the flow of protons to synthesize ATP. The ATP synthase, it catalyzes the addition of a phosphate group down here to a DP, not ATP, a DP, which is adenosine diphosphate. So we're looking for ATP, which is triphosphate, 3 phosphate groups. So we add one more phosphate, and we get the triphosphate, which is really what we're looking for. Now, all the electrons used in the electron transport chain come from NADH and FADH2 molecules. And NADH is very good at donating electrons. And the FADH2 was not as good due to the electrons being at a lower energy level. I'm not going really in depth with the process. There's 4 different pathways. This is a lot of information to digest for a person, and I have digested it. It's very complex. And I hope you can make some sense out of this as well. Ultimately, the NADH is very good at donating electrons. It's right here. So the FADH kind of skips a step, so it doesn't have as much energy. So it's not as good due to the electrons being at a lower energy level. The majority of the ATP in cellular respiration is produced in the electron transport chain. So let's talk a little bit more about ATP yield. What do we have here? What did we come up with all this? So the current estimates are that the maximum yield for a molecule of glucose, that's one molecule, is around 30 to 32 ATP. If you look it up, or you look in a textbook, you look it up online, sometimes you'll find different numbers. The current estimates, and I found this on the Khan Academy website, are 30 to 32 in an ideal conditions. So that's the ultimate kind of, if everything was working perfectly, perfect settings, no molecules were used anywhere else or shuffled out of any of the pieces of cellular respiration to be used elsewhere. If everything was just going hunky-dory perfect, we could have 30 to 32 in ideal conditions. And so we talked about glycolysis. We got 2 ATP out of that. If you remember, the citric acid Krebs cycle, we also got 2 ATP. And that was from the 2 rounds, the 2 turns of the Krebs cycle, or the citric acid cycle. Now, the electron transport chain, look at that difference. That's very significant. You could get 26 to 28 ATP, according to these current estimates for possible ATP production. So actual yields may be lower because some of the intermediates might be used in other biosynthetic pathways, so less ATP is made. This is a high estimate. Actual yields will likely and may be lower. So, quick review. We talked about the cellular respiration, 3 acts. Glycolysis, which does not need oxygen. The Krebs cycle. And the electron transport chain. We got our 30 to 32 ATP. Our cells are moving. We're feeling good. Our heart is beating. We have energy to do things. We've used this simple little sugar, a simple sugar called glucose, and we've done all of this. It's very complex. It's very interesting. It's kind of amazing that scientists could really figure this out. And thank you for listening. Thank you for watching.